Amino acids are the most basic building blocks of life.
These amino acids have many different functions. One particularly important function is its role as the building blocks of proteins.
Proteins are linear chains of amino acids. Every protein is chemically defined by its unique sequence of amino acid residues. Just as the letters of the alphabet can be combined to form a variety of words, amino acids can be linked together in varying sequences to form a vast variety of proteins. Even the simplest life generally has thousands of proteins.
These proteins are essential to organisms and are involved in virtually every process within a living cell. Proteins have structural and mechanical functions. For instance, in muscles and the cytoskeleton,
they help to maintain cell shape, while other proteins are important in cell signaling, immune responses, cell adhesion, and the cellular life cycle. One particular protein, called a motor protein, transports various cellular cargo, like energy-producing mitochondria, to cellular neighborhoods in need of fuel. They can also provide the pulling power needed to separate chromosomes during cell division. Amino acids carry information from one part of a cell to another, as well as to other cells within the organism.
So as you can see, without amino acids and proteins, basic cell structure as we know it could not exist.
Other important types of proteins are enzymes. Enzymes are catalytic proteins that have special slots which hold other molecules to make chemical reactions possible. There are over 2,000 enzymes, and each one is used to enable a chemical reaction without ultimately being altered itself.
It's been observed that the slowest biological reaction would take 1 trillion years without an enzyme, but the same reaction takes only 1/100th of a second when the enzyme is present. Pretty impressive.
Adenosine triphosphate, otherwise referred to as ATP, is the energy source the enzyme uses to enable the reaction. Living organisms require and manufacture these enzymes along with all the other proteins.
In fact, it takes over 150 existing proteins to manufacture just one protein. So in essence, it takes a protein to make a protein. It's this chicken and egg scenario that continues to baffle scientists.
When cells were discovered over 300 years ago, it was thought that the structure of a cell was very simple. With the inventions of the electron and proton microscopes, we now have a much clearer understanding of the intricacies of life. The cell is an extremely efficient and well-networked computer system with thousands or even millions of processors and billions of bits of information. Cells are the structural and functional units of all living organisms. Some organisms, such as bacteria, are unicellular, consisting of a single cell. Other organisms, like humans, are multicellular, having many cells. In fact, it's estimated that an adult human has over 100 trillion cells, and each cell is an amazing world of its own.
It can take in nutrients, convert the nutrients into energy, carry out specific functions, and reproduce itself as necessary. Even more amazing is that each cell stores its own set of instructions for carrying out each of these activities. Let's take a closer look at how a cell works.
The first thing we see is the cell membrane. Much like the firewall software on your computer, the membrane contains protein gatekeepers, allowing only those components into the cell that belong and rejects all other components. Once we pass through this membrane, we see the organelles, or the organs of the cell. Ribosomes are the computer-controlled protein manufacturers of the cell. By fastening to an mRNA and using it as a template, the ribosome arranges amino acids in the correct sequence to form a particular protein. This process is known as translation. Golgi body. Next, we find a Golgi apparatus, also known as a Golgi body. Found in both plant and animal cells, Golgi bodies package and store proteins. Much like a post office, the Golgi packages and labels items, which it then sends to different parts of the cell. It's composed of membrane-bound stacks known as cisternae. Each cisterna is made up of a flattened membrane disc, and its primary job is to modify proteins, but it's also involved in the transport of lipids around the cell and the creation of lysosomes. Lysosomes. Lysosomes are spherical organelles which dispose of all the waste in the cell. These tiny organelles contain acid hydrolase enzymes that break up waste materials and cellular debris. The membrane around the lysosome allows the digestive enzymes to work at the 4.5 pH they require to break down the debris and dispose of it. Without the lysosomes, the cell would become overwhelmed with debris and ultimately self-destruct. Mitochondria. Mitochondria are the power plants of the cell, generating most of the cellular energy known as adenosine triphosphate, or ATP. In addition to supplying ATP, mitochondria are involved in a range of other processes, such as signaling, cellular differentiation, cell death, the control of the cell cycle, and cell growth. Nucleus. The nucleus is the control center of a cell. This is where we find most of the DNA which contains the instructions needed to create other elements of the cells. Like the motherboard of a computer controlling its components, the nucleus directs the growth, metabolism, and reproduction of the cell, among other things.
Life requires fully functional DNA, RNA, ATP, enzymes, and other proteins. If any of those components were missing or not working properly, life would fail to exist.
Let's examine what DNA is and how it works to better understand how information is used in the cell.
Deoxyribonucleic acid, or DNA, is a long polymer of nucleotides, each nucleotide being a deoxyribose sugar molecule, one phosphate group, and one base. Nucleotides are joined together to form the DNA backbone. A second DNA strand then joins the first to form a double helix. You may recognize the code letters A, C, G, and T, which are the 4 bases used in DNA. These bases are adenine, A, cytosine, C, guanine, G, and thymine, T. Like the code in Mr. Gates' software, the sequence of the 4 bases encodes genetic instructions used in the development and functioning of all known living organisms. Once organized, this information is what makes up the gene. And genes, as we all know, contain the recipes for the components of life. Genes hold all the information to build and maintain an organism's cells, and they also pass genetic traits to offspring. Human DNA contains an estimated 20 to 25,000 genes in its approximately 3 billion base pairs.
And did you know that if the DNA in an adult human's body were laid out end to end, it would be about 30 billion miles long? That's enough to travel the distance from the Earth to the Sun over 320 times. A gene contains both coding sequences that determine what the gene does and non-coding sequences that determine when the gene is active, as well as thousands of other regulatory functions. Less than 2% of the genome is for coding proteins. The other 98%, which used to be thought of as junk DNA, is now being studied to determine its functionality.
Chemically, the DNA in the cells of my body is the same as that in a fruit fly, an orange, a hippo, an elephant. It's all the same chemically. The uniqueness to all of those species of living things lays in the information content of the DNA. So while the chemistry may be the same, the actual information that's embedded in that chemistry is very, very different. And the challenge for molecular biologists such as myself.
But we have proteins that are actually creating the DNA molecule. That's our blueprint. And yet on the DNA molecule, there are sequences, a part of the DNA that are coded for those proteins. So the manufacturing plant needs the blueprint, and the blueprint, the DNA, needs the manufacturing plant in order to actually create the DNA. So what came first, the proteins that made the DNA or the DNA that made the proteins?
Once DNA is organized into a single coiled piece,
It's then wrapped around a complex hierarchical structure of proteins. This is what is commonly known as a chromosome. There are 23 pairs of chromosomes in the human genome. A genome is a full set of chromosomes containing all the inheritable traits of an organism. And as we observe our surroundings, we can see there are many different genomes in existence, creating very unique and incredible organisms.
If it seems like your body is constantly working hard, it is.
Living organisms continually produce new cells through a process called cell replication throughout their entire lives. And here's how it works. First, we start with our DNA double helix. Helicases are molecular motors that use the chemical energy of ATP to break hydrogen bonds between bases and unwind the DNA double helix into single strands.
This process is used for both replication and repair of the strands. If unwound, a single DNA strand would be about 6 feet in length. And yet, it fits into a cell which can't even be seen with the human eye.
Remarkably, these strands are kept from tangling as they are separated and duplicated. Next, these strands are copied by enzyme-based computers, creating an exact replica of each original strand. Over 30 different proteins are required for duplication and repackaging of cellular components during cell division. It's from this tiny, masterfully complex cell that life is formed. All living organisms begin with one cell. As cell division takes place, we see multicellular organisms take form. When we stop to think about our own body having approximately 100 trillion cells, each group having a unique purpose, such as epidermis, the lungs, heart, skeletal structure, muscles, nervous system, digestive system, reproductive system, immune system, and the brain all working simultaneously to form this magnificent machine we call the human body, we begin to have an appreciation for the complexity, elegance, and beauty of life.
You know, we're all derived from a single cell, the zygote. And what marvels me is from this single cell, we get 250 distinct cell types, each of which has a very, very unique program. All of the cells, with a couple of exceptions, have a nucleus and have a genome. And that genome is the same across all the cells. So when we begin to think about the complexity of the genome, I think that's amazingly complex, how we go from a single cell to unique cells that have very specific programs of gene expression that are necessary for that unique cell to function. But we're only beginning to understand the information in our genome that directs those unique programs of gene expression.